Hello, my lovely students. I hope you all are doing well. So, I'm Anchal Rohila, and today we are going to start with a recap of a very important chapter that is biological classification. It is one of the important chapter in which we study about your bacteria, protista, fungus, and some details about the viruses. So, without any delay, let's start with today's session. So, first we'll start with the five kingdom classification, right? So, there were various system or classifications given by various people, right? So, we are going to first start about the five kingdom classification. Five kingdom classification. It was given by R.H. Whitaker. Five kingdom classification was given by R.H. Whitaker, right? There were two kingdom classification which was given by Linnaeus. Linnaeus divided the organism into Planta and Animalia, mostly on the basis of their cell wall. If cell wall is present, it's plant. If cell wall is absent, it is animal. So R.H. Whitaker gave the five kingdom classification on the basis of what is their cell type, whether it is prokaryote, eukaryote, what is their structure, body structure, whether it is made up of one cell or many cells, or tissue is formed, organ is formed, and type of reproduction, type of nutrition, and also evolutionary relationship, which means from which ancestor they came or they evolved. So five kingdoms were Monera, Protista, Fungi, Plantae, and Animalia. These were the five kingdom that was given by R.H. Whitaker. Next is, first, what about their cell wall? A cell type. So Monera, because it includes bacteria, all the bacteria on this earth is included in Monera. They are prokaryote, otherwise Protista, fungus, Plantae, and Animalia all are eukaryotes. I've already told you the difference between prokaryotic cell and eukaryotic cell. Organism having prokaryotic cell are known as prokaryotes. Organism having eukaryotic cell are known as eukaryotes. Let's talk about their cell wall. Beta, here in bacteria, cell wall is non-cellulosic, which means it is not composed of cellulose. It is composed of polysaccharide and amino acid and known as peptidoglycan, right? So cell wall of bacteria is known as peptidoglycan. I'll talk about it. Protista, the cell wall is present in some member and absent in some. Fungi, cell wall is made up of chitin. Plant cell wall is made up of cellulose, and in animal it is absent. Okay. Next, nuclear envelope. Whether the membrane is present around the genetic material or not. In Monera, it is absent. That's why it is the prokaryotic cell. Whereas in Protista, fungi, plant and animalia, because they are eukaryotes, which means their genetic material is protected by the envelope. Body organization. Monera, cellular, which means they are made up of single cell. Protista, they are also made up of single cell. In fungi were the first multicellular organism, but loose tissue are present, which means they don't form a proper tissue. Although many cells are present in the body of fungi, but they don't form a proper tissue. Whereas a proper tissue was first formed in Plantae, and in some plants, organ is also seen. Like organ is like stem, root, and leaf. They are the organs of plant. And in Animalia, you can see animal at tissue level, organ level, and organ system level, right? So as you can see, Monera and Protista are only made up of one cell. Let's talk about their mode of nutrition. Beta, Monera can be autotrophic or they can be heterotrophic, right? But mostly bacteria are heterotrophic. So bacteria can be for autotrophs, they can make their own food, or heterotrophs, they are dependent on others for their nutrition. Most of the bacteria, they depend on others to get the nutrition. Same in Protista. They can be autotrophs, they can be heterotrophs. So both type of categories are seen in Protista. Some member of Protista are autotrophs and some are heterotrophs. Fungi, they are strictly heterotrophs beta. Plants, they are strictly autotrophs. Autotrophs means they can make their own food. Heterotrophs means they are dependent on other for their nutrition. And animalia are strictly heterotrophic, which means animals, they feed on others to get their nutrition. So this type of comparison was given by R.H. Whitaker, right? That how all these five kingdoms are different from each other. In this chapter, we are going to include Monera, Protista, and fungi. Right? In the next chapter, we will be studying about Plantae, and in zoology, you will be studying about Animalia. Right? So I hope till here everything is clear, because everything we have already studied in our lectures. So without any delay, let's start with kingdom Monera. I'll give you some brief of Monera. So kingdom Monera basically includes all bacteria. It includes all bacteria, and they are unicellular and prokaryotes. They are unicellular and prokaryotes, which means they are made up of one cell and they are prokaryotes. And kingdom Monera is divided into two groups: Archibacteria and Eubacteria. Monera is divided into two groups, Archibacteria and Eubacteria, mainly on the basis of cell wall and cell membrane differences. So there are some differences in cell wall and cell membrane between Archibacteria and Eubacteria. Archibacteria and Eubacteria. That's why kingdom Monera is divided into two groups, Archi and Eubacteria, because of their difference between their cell wall and cell membrane. Okay, so all the bacteria on this earth included under kingdom Monera. They are prokaryotes. They are the only prokaryotes. Let's first discuss about Archibacteria. Better remember that Archibacteria, they live in extreme conditions. They like to live in extreme condition, which means like extreme temperature, extreme pH, extreme salty condition. So there are three types of Archibacteria: methanogens, halophiles, and thermoacidophiles. As you can see, methanogens, they produces methane gas. That's why they are known as methanogens. They produce methane gas. They are found in marshy areas. Marshy means where the water content in the soil is high. Marshy area, and they are also found in the gut of ruminant animals like cow. So they are also live inside the gut of cow, buffalo. They are chemoautotrophs beta. They are chemoautotrophs. Chemo means chemical energy. Autotrophs means to synthesize food. So chemosynthesis means synthesis of food using chemical energy. Synthesis of food using chemical energy. Let's come to halophiles. Beta, halo term means salt. Halo means salt, right? So here halophiles means they live in extremely salty condition, and they are heterotrophs, which means dependent on others for nutrition. They are dependent on others for nutrition. Thermoacidophiles. Thermo means high temperature, and acido means low pH. So the bacteria that are found in high temperature and low pH, high temperature approximately 80 degrees Celsius and low pH approximately 2, those bacteria are known as thermoacidophiles, and they are also chemoautotrophs, which means they synthesize their food with the help of chemical energy. Right? So Archibacteria are three types: methanogens, halophiles, and thermoacidophiles. This is a very important thing to remember. I hope everything is clear. After this, we'll study about Eubacteria. Beta, Eubacteria means they are the true bacteria. They are the true bacteria. They are the typical bacteria. Cell wall is made up of peptidoglycan. Cell wall is made up of peptidoglycan. The cell wall of Eubacteria are made up of peptidoglycan. Peptidoglycan means sugar plus amino acid. Sugar plus amino acid is peptidoglycan. Okay. So first Eubacteria that we are going to study is cyanobacteria, beta. Cyanobacteria is also known as blue-green algae. Why? Because they have a chlorophyll A similar to green plants. Right? Blue-green algae does not mean that it's a plant. Blue-green algae, it's a bacteria. It's a bacteria. Why we are calling it blue-green algae? Because it has some chlorophyll similar to the plants. They are photosynthetic autotrophs, which means synthesis of food with the help of light energy. They synthesize food with the help of light energy, and beta, they can be unicellular, colonial, or filamentous, which means when they are present as a single cell, it is unicellular. When many cells present like this, close to each other, which means they are forming a colony. And when cells are placed over each other, this is known as filamentous. Colonial and filamentous does not mean that they are multicellular, right? In colonial and filamentous, each cell is capable of doing its own function, but they like to live in colony. They like to live like filamentous condition, right? But in colonial and filamentous, each cell can do their own function. They are capable they are capable to do their own function. But the only thing is they like to live in colony or filamentous condition. Okay. Now they can be found in fresh water, marine water, or terrestrial conditions. So cyanobacteria can be found in fresh water, marine, or terrestrial condition. They form algal bloom in polluted water. Beta, algal bloom means excessive growth of cyanobacteria in the water. So have you seen a green mat-like structure on a water body? That green mat-like structure over a water body is algal bloom. Why algal bloom is formed? Due to excessive growth of cyanobacteria. Example of cyanobacteria are nostoc and anemone, very important examples. And as you can see, this is the filamentous form, nostoc. This is the filamentous form of cyanobacteria, nostoc. And you can see the filamentous is protected by or covered by a mucilaginous sheath. And there is a special cell, heterocyst. What is the role of this special cell, beta? The role of Heterocyst is a special cell, is a conversion of nitrogen into ammonia. We call it atmospheric nitrogen fixation. We call it nitrogen fixation. When nitrogen is converted into ammonia, it is known as nitrogen fixation. Why cyanobacteria converts nitrogen into ammonia? Beta, plants, they need nitrogen and they cannot use nitrogen from the atmosphere. So plants, they take help of the bacteria to convert atmospheric nitrogen into ammonia so that they can use the ammonia form to synthesize various compounds like DNA, RNA, amino acid, hormones, vitamins. Every compound contains nitrogen here. So yes, these bacteria are basically helping the plant to get their nitrogen, right? And heterocyst is a special cell in cyanobacteria that can do that. Okay, so I hope everything is clear till here. Next bacteria are Mycoplasma. Next bacteria are Mycoplasma, beta. Mycoplasma, they are the smallest cell or bacteria. They are the smallest cell or bacteria. There is no cell wall, which means they are wall-less bacteria. They don't have any cell wall. So Mycoplasma is an example of bacteria having no cell wall. They are non-motile, which means they don't have flagella. Remember beta, cyanobacteria also don't have flagella. Cyanobacteria lack flagella. They are also non-motile. Flagella is a tail-like structure that helps in movement. Cyanobacteria don't have flagella. Mycoplasma don't have flagella. They are highly pleomorphic. Pleomorphic means can change their form. Because they don't have cell wall, so they can change their shape. They can be filterable through bacterial filter because they are smaller than other bacteria. So they can easily be filterable through bacterial filter because they are even smaller than their fellow bacteria. They are resistant to penicillin antibiotic. Why? If we want to kill Mycoplasma and if we give penicillin to Mycoplasma, Mycoplasma won't die. They are resistant to penicillin antibiotic. Why? Because beta, penicillin acts against cell wall. Penicillin acts against cell wall. And Mycoplasma lack cell wall. Okay, so that's why if we give penicillin to Mycoplasma, then Mycoplasma won't die, right? Because Mycoplasma don't have cell wall and penicillin act against a cell wall. This is how a cell of Mycoplasma look like. This is how the Mycoplasma bacteria look like. This brown layer is lipoprotein, which is nothing but membrane. Membrane, as you know, plasma membrane is made up of lipid and proteins. This is DNA, ribosomes, RNA. So this is how a cell of Mycoplasma, single cell of Mycoplasma looks like. Okay, so this Mycoplasma is the another example of Eubacteria. In our syllabus, we need to learn about cyanobacteria and Mycoplasma. Okay, and remember this Mycoplasma is parasitic to both plant and animal. They can cause infection to plant and animals as well. And Mycoplasma is basically can survive in the presence of oxygen or without oxygen. So they can live in both condition, whether the oxygen is provided or not. So Mycoplasma can live without oxygen or can survive without oxygen, right? So Mycoplasma, they can adjust, they can make compromise, right? If oxygen is there, they are happy, but if oxygen is not there, they are still happy, right? So if we take out the oxygen from Mycoplasma, they won't die, right? They can survive without oxygen. If oxygen is there, it's okay. If oxygen is not there, still they can survive, remember, right? What is the role of oxygen in a cell? The role of oxygen is in the respiration, right? Oxygen helps in respiration. So yes, Mycoplasma is basically your smallest cell. This is where we have completed the kingdom Monera. Now we'll start with kingdom Protista. So beta, kingdom Protista includes unicellular organism which are eukaryotes, right? So kingdom Protista includes unicellular eukaryotes. So all the unicellular eukaryotes come under kingdom Protista. So eukaryotes for the first time came under kingdom Protista. Let's discuss first group of Protista, that are photosynthetic protist, which means that can synthesize food with the help of light energy. That can synthesize food with the help of light energy, right? So there are three diatoms, dinoflagellates and euglena, right? But diatoms and desmids, they are the two organisms that come under a group known as chrysophytes. Remember, there's a group known as chrysophytes in which diatoms and desmid come. We need to understand only about diatoms. So think like this, that diatoms are the main hero here and desmid are like doing the side role. So we need to understand about diatoms. So I always write it in a tabular form so that you can compare all the organism. Let's talk about their habitat. Habitat means their home. Where do you find these organisms? Diatoms can be found in fresh water or marine. They float passively in the water current. So they can move in the direction of water current, right? They will move with the water current. Dinoflagellates are mostly marine. They are found in marine water. And euglena, most of them are fresh water. Let's talk about their cell wall. Diatoms, the cell wall is made up of silica, not easily destructible, very strong, cannot be degraded so easily. Right? And the cell wall of dinoflagellates having cellulose and also pectin, but mostly cellulose. In euglena, beta, cell wall is absent, remember. In euglena, cell wall is absent. Instead, they have a protein layer known as pellicle. Remember, this is very important that euglena have a proteinaceous layer known as pellicle. Let's talk about their photosynthetic pigments because they can do photosynthesis. So yeah, they have photosynthetic pigment. They have chlorophyll A, C, beta carotene, xanthophyll. Beta chlorophyll are green pigment. Xanthophyll are generally yellow pigment and carotene are generally orange pigment. Dinoflagellates contain chlorophyll A, C, beta carotene, alpha carotene, xanthophyll. They all help in photosynthesis. Euglena has chlorophyll A, B, beta carotene and xanthophyll. Chlorophyll, carotene and xanthophyll all together involved in photosynthesis. Reproduction beta, mainly asexual reproduction. Mainly diatoms can do asexual reproduction, but they can also undergo sexual reproduction. Dinoflagellates also mainly asexual reproduction, but and can also do sexual reproduction. Euglena beta, they do binary fission, which is a type of asexual reproduction. But in Euglena, sexual reproduction is not known to occur. In Euglena, sexual reproduction is not known to occur. They can do only binary fission. Okay. But diatoms and dinoflagellates can do both. I hope everything is clear. After this, let's talk about their flagella. Beta, flagella is absent in diatoms, but they can have flagella in reproductive stage like in gametes. Gametes can have flagella in diatoms. Dinoflagellates at the name indicate dino, two, flagellate, flagella. They have two flagella. They have two flagella, heterocont. Heterocont means different. They have two different flagella. Euglena have two flagella. One is very long and other is very short. Euglena also has two flagella, but one is long and another is short. So this is how diatoms look like. Their structure is soapbox like structure. Their structure is soapbox like structure, and this is dinoflagellates, and this is how Euglena look like, right? Beta, as I told you that diatoms, the cell wall is made up of silica, and silica is very difficult to digest or destruct. So when diatoms die, when these diatoms die, right, all the thing get digested except silica, and over the years, hundreds of years, I'm talking about hundreds of years, over the hundreds of years, their silica get deposited, and after that hundreds of years, you will get a product from mining known as diatomite or your diatomaceous earth, right? So deposition of silica over years form diatomaceous earth. It's a powder structure. It's like a powder which we can use for polishing and which we can use for filtering syrups, because that powder is somehow having rough consistency or rough structure. That's why diatomaceous earth can be used for polishing and some filtering the syrups or any other liquid. Okay. Dinoflagellates, if I talk about, they cause red tide beta. They make the water looks or appear red in color due to excessive growth of dinoflagellates like gonolex, right? So excessive growth of this dinoflagellates, gonolex, can cause red tide. It makes the water red, appear red in color due to excessive growth of dinoflagellates like gonolex. I hope this is clear to all of you. Talking about Euglena beta, Euglena is photosynthetic in presence of sunlight, but if sunlight is not there, they become heterotroph. So they can do both. When sunlight is there, they are photosynthetic. If sunlight is not there, they are heterotrophic, right? They can do both. In the presence of sunlight, they are photosynthetic. In the absence of sunlight, they are heterotrophic, right? I hope this is clear to all of you. Let's talk about slime molds. Right? Slime molds are basically decomposers, right? They are saprophytes. They are saprophytes, beta. So what happens to slime molds? Under favorable condition, when they have lots of food and other favorable condition, they form a structure like this, a big cell containing many nucleus. So under a favorable condition, they form a big cell containing many nuclei. This structure is known as plasmodium. This is plasma membrane. These are nuclei. Right? What happens now? The unfavorable condition comes. Unfavorable condition comes. And they form a body known as fruiting body, beta. They form a body known as fruiting body. And inside fruiting body, you will have spores. These are spores and this is fruiting body. Right, so under unfavorable condition, they form spores. Spores look like this. They have cellulose cell wall. That's why they are known as true spore, because they have a wall of cellulose. They are dispersed by air. They are dispersed by air from the fruiting body. So air will disperse the spores. Wherever spores find a favorable condition, it will form the male gamete and female gamete. So whenever the spore finds favorable condition, it forms male and female gamete. Both will fuse and form zygote. And again, the same cycle continues. Again, the same cycle continues. So yes, this is how slime molds look like. I hope this is clear to all of you. This is we have already discussed in our main lectures. Okay. So after slime mold, there's a last group of protista known as protozoans. Protozoans are of 4 types. They are also saprophytes. They are also saprophytes or parasitic. Or we can commonly write they are heterotrophs. Right? They can be saprophytic, which means can feed on dead or decay, or they can be parasitic, which means take their nutrition from the living matter. Amoeboid, flagellated, ciliated, and sporozoans. Let's talk about their habitat. Beta, amoeboid are found in fresh water, sea water, or moist soil. So amoeboid can be found anywhere. Flagellated protozoans, they can be free living. They can live without any organism, or they live with other organism. Parasitic means when they cause infection to the organism. Ciliated are aquatic, and sporozoans are endoparasite, which means they live inside host and cause infection. Endoparasite means those parasites that live inside host and cause infection. Let's talk about their locomotory structure. Which structure helps in their movement? Amoeboid having pseudopodia, beta. Pseudopodia is like false feet. Right? This is what we call pseudopodia. Right? This is amoeba. So this pseudopodia helps in the engulfing of food and also helps in the movement of amoeba. Flagellated protozoans have flagella for their movement. Ciliated protozoans have cilia, and in sporozoans there is no such locomotory structure. Okay. Special feature, in amoeboid you have silica shell, right? You have silica wall in some members. In flagellated protozoans, rarely you can see any reproduction. They generally do asexual reproduction. Ciliated protozoans have a defined region of ingestion and egestion, which means ciliated protozoans have mouth and anal opening. Their mouth is known as cytostome and their anal opening is known as cytopyge. This is, you have already, I think, studied in your 9th standard, the structure of paramecium. They have proper mouth and cytopyge. In sporozoans, infectious spore produce, which causes infection, right? Like plasmodium. Plasmodium causes malaria. It causes malaria. Beta, this is different from this plasmodium. This plasmodium is a structure formed in slime mold, and this plasmodium is a sporozoans that causes malaria. Example of ciliated protozoan is paramecium. Example of flagellated is trypanosoma. Everybody knows that it causes sleeping sickness. It causes sleeping sickness. Amoeboid protozoans, amoeba and ant amoeba. Ant amoeba causes dysentery. It causes dysentery. Okay? So this is about protozoans. Here we finish our kingdom Protista. We'll be starting with kingdom Fungi. I hope everything is clear till here. Fungi, beta, are multicellular, except yeast. Yeast is a unicellular. Yeast is unicellular. Okay? And they are eukaryotes. They are multicellular eukaryotes. According to their body, mycelium is the body of fungi. According to their spore, what type of spore is formed and what type of their fruiting body, they are divided into various classes: Phycomycetes, Ascomycetes, Basidiomycetes, and Deuteromycetes. Okay. NCRT has not mentioned this, but I mentioned this. Phycomycetes is further divided into two groups: Oomycetes and Zygomycetes. Right? Let's start with their common name. Common name of Oomycetes is known as algal fungi. Why we call it algal fungi, I will tell you later on. Zygomycetes is known as conjugation fungi. Ascomycetes is known as sac fungi. Basidiomycetes is known as club fungi, and Deuteromycetes are known as fungi imperfecti. Why we call Deuteromycetes as fungi imperfecti? Because sexual reproduction is absent in the member of Deuteromycetes. That's why we call them imperfect fungi. That's why we call them imperfect fungi. Habitat, beta, only Oomycetes are found in aquatic condition. Otherwise, all fungi are found in terrestrial condition. All fungi are found in terrestrial condition. The cell wall of Oomycetes is made up of cellulose. Remember, only one group of member of fungi where the cell wall is made up of cellulose. These are the reason why it is known as algal fungi, because these two feature resembles with the plant. Otherwise, all other fungi have their cell wall of chitin. Hyphae. Both Oomycetes and Zygomycetes have a cenocytic hyphae. Cenocytic hyphae means when they don't have any septum between their nucleus. When the body is like this is known as cenocytic. Whereas Ascomycetes, Deuteromycetes, and Basidiomycetes have septate body, which means septate means like this, where you will find septum after each nuclei. This type of body is known as septate, and this type of body is known as cenocytic. Right? So both the members of Phycomycetes, Oomycetes and Zygomycetes have cenocytic body, whereas Asco, Basidio and Deutero have septate body. Okay? I hope this is clear to all of you. Next is how do they reproduce asexually? Okay? Oomycetes and Zygomycetes beta Oomycetes asexual spore is zoospore, which is a motile spore. It can move, whereas Zygomycetes reproduce asexually through sporangiospore, which is a non-motile spore, also known as aplanospore. Aplano means without flagella. Ascomycetes reproduce through conidia, which is also a non-motile spore. In Basidiomycetes, asexual reproduction is generally absent. In Deuteromycetes, it is also through conidia. It is also through conidia. Let's talk about sexual reproduction. Beta sexual reproduction through oospore. In Oomycetes, through zygospore in Zygomycetes. Through ascospore in Ascomycetes. What are ascospore, beta? This is ascus, and inside ascus, ascospores are formed. Inside ascus, ascospores are formed, and each ascospore can give rise to new individual. Right? Ascus is a sac-like structure. That's why they are known as sac fungi. That's why they are known as sac fungi. Sac means bag. Ascus is a bag-like structure. Let's talk about Basidiomycetes. They have a club-shaped structure. They have a club-shaped structure over which you will find basidiospore. This is basidiospore, beta. When the basidiospore release from the parent body, it can give rise to new fungi. This is basidium, which is a club-shaped structure. That's why it is known as club fungi. Ascomycetes is known as sac fungi because it has a sac-like structure known as ascus. It is a club fungi because it has a club-shaped structure known as basidium. And as you know, in Deuteromycetes, the sexual reproduction is absent. The sexual reproduction is absent. This table will surely help you, right? When you read from NCERT, you will get every point because I have written every point here from the NCERT. Here are some of the disease caused by fungi, like this is rust disease. As you can see the rusty structure here, by rust fungi known as Puccinia. Rust fungi is Puccinia. This is white rust, beta. This is white rust by Albugo. This is white rust caused by Albugo, and this is smut disease where the grain become black by smut fungi. By smut fungi Eustylego. By smut fungi Eustylego. So here are some of the disease caused by fungi. Rust fungi, white rust and smut disease. I hope everything is clear. There is one more topic left about the virus. Right, let's talk about the virus here, beta. Virus is an acellular entity. Virus is an acellular entity. They are the connecting link. They are the connecting link between living and non-living, because they have some feature of living organism and some feature of non-living things. Viruses have three things in their body: envelope, capsid, and genetic material. They have three things in their body: envelope, genetic material, and capsid. Envelope is made up of lipid, protein, and carbohydrate. It may or may not present. So if presents, it's not important. It may or may not present. So it is not necessary to be present in all viruses. Let's talk about capsid, beta. Capsid it is a protein layer that protects the genetic material. It is a protein layer that protects the genetic material, and it is present in all virus. Genetic material, it is either DNA or RNA. It is either DNA or RNA. Both are not present in same viruses. There are some subviral agents. There are some subviral agents that causes infection, like your viroids. Viroids are infectious RNA molecule without protein layer or without capsid. They are the infectious RNA molecule without the capsid. And second is prions. P for prions, P for proteins. They are infectious protein molecule. Prions are the infectious protein molecules that causes infection. Viroids are only RNA and prions are only protein that can cause infection. Okay. I hope this is clear to all of you. Here are some of the viruses. Beta, this is the tobacco mosaic virus. Some of the early discoveries were done with the help of tobacco mosaic virus. They have a genetic material RNA, and this is capsid. Capsid is made up of single unit known as capsomere. Capsid is a layer that is made up of single unit known as capsomere. This is beta bacteriophage. Remember, tobacco mosaic virus, the host is plant. It causes infection to plant. Whereas bacteriophage, the host is bacteria. The host is bacteria. They causes infection to bacteria, and they have DNA as a genetic material, and this is capsid. They have DNA as a genetic material, and this is capsid. So these are the two viruses that are in your syllabus: TMV, tobacco mosaic virus, and bacteriophage. Okay. So before we end this lecture, let's solve some of the questions. Which of the following is a wrong statement? Diatoms are the chief producer of the ocean. Yes, they produces food, they synthesize food. Diatoms are microscopic. Yes, they are made up of only single cell. They can only be visible through microscope, and they float passively in water. Yes, they will go with the water currents. The wall of silica is easily destructible. No, not so easily, because wall is made up of silica. Wall is made up of silica. This is wrong statement. Diatomaceous earth is formed by the cell wall of diatoms. Yes, I told you, whenever the diatoms die, their silica get deposited, and over the years we will get a product known as diatomaceous earth. So third option will be our answer. Third statement is incorrect. The wall of diatoms are not easily destructible. Nuclear membrane is absent in. Beta, they are talking about prokaryotic cell. They are talking about prokaryotic cell. Nostoc is a prokaryotic cell because it is a cyanobacteria. It is a cyanobacteria. Penicillium is a fungi. Volvox is a algae. And agaricus is a mushroom, which is also a fungi. All other are eukaryotes except nostoc. And the last question is pigment containing membranous extension in some bacteria. So this is the plasma membrane, and in some bacteria, plasma membrane extend towards the cytoplasm and contains some pigment. This body is known as chromatophores. This body is known as chromatophore. They are formed in photosynthetic bacteria, found in photosynthetic bacteria. So those bacteria which do photosynthesis, in those bacteria you will find chromatophore. Okay, I hope everything is clear. This is the kind of revision you want, because every detail we have studied in our lectures. All the concepts are brushed up now. Okay, so I hope you really enjoyed the lecture. I'll see you in the next time, or I'll see you next time. Until then, take care everyone. Bye.